Hello, everyone, and welcome to today's webinar hosted by Organovo and presented by Leah Naroda, doctoral candidate at the University of North Carolina, Chapel Hill. My name is Jeff Ireland. I'm Director of Scientific Applications here at Organovo. Before we get started, I want to remind everyone in the audience that we will address your questions at the end of the presentation. We encourage you to submit your questions as they come to you during the presentation by typing them into that questions pane on the control panel to the right of your screen. We will collect the questions and address as many as we can during the Q&A section. With that, it's now my pleasure to introduce Leah Naroda. Leah received her BS in pharmacology from the State University of New York at Stony Brook and is currently pursuing her PhD in toxicology at the University of North Carolina at Chapel Hill under the guidance of Dr. Paul Watkins and Dr. Mary Mosdale. As you'll see today, Leah's been doing really innovative work with our bioprinted liver tissue, applying it to the very challenging problem of accurately modeling hepatic fibrosis in an in vitro system. So I'll turn it over to you, Leah.
Awesome. Thank you, Jeff, for the introduction. And I'd just like to take a moment to thank the organizers for the opportunity to speak to you today. For those of you who tuned into the first presentation of the webinar series, Dr. Sharon Presnell gave a wonderful overview of Organovo's 3D bioprinted human liver tissues. And today I'm going to talk more about the utility of this system to model more complex processes such as compound-induced liver injury leading to fibrosis. And I just wanted to remind you that why DILI is still a serious concern. And it's not only an issue faced in the clinical setting, but also the pharmaceutical industry, and often involves a number of complex mechanisms and series of events, which are shown here on the left-hand side. And because of this, DILI is often difficult to detect in preclinical species and clinical trials. And here at the Institute for Drug Safety Sciences, we implement innovative approaches to better understand mechanisms to predict toxicity and prevent adverse reactions to drugs. In my case, I'm particularly interested in the events that occur in the setting of recurring injury. But before I get into hepatic fibrosis, I just wanted to illustrate why this type of injury response poses a challenge for toxicity risk assessment using methotrexate as an example. So methotrexate is an antifolate commonly used as an anti-neoplastic agent, but is also effective at low doses for extended periods of time for the treatment of inflammatory disorders such as rheumatoid arthritis or psoriasis. And while the more common side effect is elevated blood levels of alanine aminotransferase, a fraction of patients actually go on to develop cirrhosis and end-stage liver failure in more extreme cases if the agent is not removed. And these features make it difficult to detect before it is relatively well advanced. Now, one of the hallmark features of this response is fibrosis, which typically occur under these conditions of recurring low exposure to an agent. So what is fibrosis? Fibrosis is essentially an abnormal wound healing response, which is illustrated in these cartoons of both normal and fibrotic liver. So typically in the setting of recurring injury to the parenchyma or the hepatocytes leads to the presence of myofibroblasts, which are the major collagen-producing cell in the liver. Now these myofibroblasts are derived from activated hepatic stellate cells shown here, which exhibit a quiescent phenotype in an uninjured liver. And these cells are primarily responsible for the collagen accumulation and scar tissue formation. In addition to this, you have chronic inflammation illustrated here with the activation of resident macrophages or Kupffer cells and the recruitment of extrahepatic inflammatory cells to the site of injury. And now this sustained production of growth factors and also pro-fibrogenic cytokines ultimately drive this response and leads to a net increase in collagen deposition and an overall disruption of normal tissue architecture. So because fibrosis is a complex and multifactorial process, in order to translate this knowledge into a paradigm that's useful for toxicity risk assessment remains a challenge. And so an attempt to build a structured approach to be able to assess the fibrogenic potential of drugs and chemicals, the Joint Research Center proposed the following adverse outcome pathway, which shown here is a conceptual framework that is built on the integration of mechanistic information from in vitro, in vivo, and in silico systems to benchmark risk assessment and regulatory application. And now, while many of the key events strongly associated with fibrosis have been elucidated from animal models, the key initiating and series of adaptive events that perpetuate this response in humans is still not well understood. So in order to better understand the roles of the relative cell types involved in this response in a systematic fashion, an in vitro approach is warranted. However, there exist a number of limitations of conventional model systems to study some of these more complex processes, such as fibrosis. The first being that they're relatively short-lived and greatly hinder our ability to perform some of this chronic low-dose exposure modeling. Furthermore, these models are too simple and often lack relevant cell-cell and cell microenvironment interactions, which play a key role in mediating the liver's response to injury. And lastly, the constitutive activation of stellate cells, shown here in the bottom series of photomicrographs, where stellate cells transdifferentiate into myofibroblasts in standard 2D models over time, greatly confounds our attempts to study the direct compound effects on the activation of this cell type. And as I had mentioned before, these are the primary cell types that produce the collagen scar following hepatotoxic insult. Furthermore, this heat map on the lower right-hand side representing gene expression profiles of culture-activated hepatic stellate cells versus stellate cells that have been activated in vivo as a result of bile duct ligation and carbon tetrachloride treatment, which are two well-known methods of inducing fibrosis in rodents, demonstrates the need for model systems to accurately recapitulate this cellular phenotype as stellate cell activation would occur in vivo. As such, we propose to take a novel three-dimensional approach to dissect some of the key initiating and series of adaptive events leading to hepatic fibrosis using Organovo's 3D bioprinted liver model. And so this is a picture of the NovoGen MMX bioprinter, and this is what is used to create 3D bioprinted tissues. Now, bioprinting is an automated process by which these multicellular tissues are formed. And so if you think about standard 3D printing, instead of using a polymer or plastic, your ink is now essentially cells in a hydrogel that are printed layer upon layer to build a three-dimensional structure. So the hydrogel portion of the bioink acts to hold everything together in the initial fabrication of the tissue, but dissipates once the tissues are cultured as the cells coalesce into a tissue-like structure. And the XV 3D bioprinted liver is comprised of three primary cell types of human origin, including hepatocytes, hepatic stellate cells, and endothelial cells. Now, the advantage of using this bioprinted process is to create these tissues is that it's an efficient and reproducible means of establishing some of the key architectural relationships between cells. And when you culture these cells in a 3D context, it more accurately resembles a tissue-like cellular density, heterotypic parenchymal and non-parenchymal contact shown here with vimentin staining, and polarization of epithelial cells shown here with E-cadherin. And so on the top right-hand corner, this is a cartoon of bioprinted liver sitting on a transwell insert. And one of the advantages of printing, bioprinting over other three-dimensional models such as spheroids is that it affords precise control over the tissue fabrication process, and that you can further define compartments within the tissue, shown here with CD31 to highlight the non-parenchymal compartment, and here in red with albumin to highlight the hepatocellular compartment or the parenchymal compartment. Furthermore, the substantial biomass available enables histological assessment of these tissues. And this remains a gold standard for the accurate detection and assessment of liver fibrosis. So because of the multicellular nature and key architectural relationships that exist between cells in a 3D context, this provides support for extended viability and function. And here are just graphs showing that normal liver function over a span of 42 days, shown with albumin and transferrin. And these black arrows denote where typical 2D cultures begin to decline in viability and function. And this is further shown in these graphs on the left-hand side, where you have, in the gray bars, you have the 3D bioprinted liver model, and then the black bars show two-dimensional liver models that there's sustained ATP content and function over time compared to 2D models. And this longevity and retention of liver-specific function makes it a unique platform for evaluating chronic exposure to potential fibrogenic agents. In addition, the metabolic competence of the tissues illustrated here on the lower right-hand side with CYP3A4 mediated metabolism of the substrate midazolam to hydroxymidazolam in the presence and absence of the CYP3A4 inducer, rifampicin, out to 28 days is critical to study compounds that might undergo bioactivation to a toxic metabolite. Furthermore, stellate cells in this three-dimensional context regain a quiescent-like phenotype, which is shown here in the series of panels. So here on the left, we have stellate cells in 2D culture, and the brown staining represents alpha smooth muscle actin, which is an activation marker. And when these cells are incorporated into bioprinted tissues, you start to see an absence of brown staining, suggesting that the cells are in a quiescent-like state. You could further go on to take these cells out of the tissues and culture them in 2D again and find that they undergo culture activation. This is further supported with Desmond staining, which is a marker of hepatic stellate cells, and alpha smooth muscle actin, which I had mentioned before, is the activation marker. And you can see that within the tissues, a majority of the cells are Desmond positive, suggesting that they are indeed in a quiescent-like state. And so this activation process is critical to fibrogenesis and is one of the key steps before you start to see the synthesis and deposition of collagen. And so this feature uniquely enables the model to be used in the assessment of compound-induced effects on some of these early fibrogenic processes. And this is something that has not been feasible to date using some of the conventional 2D approaches. So I stated that there's a need for more biologically relevant in vitro models to assess liver toxicity, particularly in the setting of chronic exposure. And we propose to use this 3D bioprinted model system, which to us exhibits key criteria for modeling fibrosis, and that is multicellular, preserves tissue architecture, and is functional and long-lived, and preserves the phenotypic features of hepatic stellate cells. So we wanted to see whether or not we could mimic the onset of compound-induced fibrosis. So if you remember back to the adverse outcome pathway, given that we have some of the key cell types involved in the process represented in the tissue, such as the hepatocytes, which are the proposed target cell population, but also some of the stromal support cells, particularly the hepatic stellate cell, which are major effector cell types in their response, we hypothesized that we would see a robust fibrogenic response as a result of repeated low concentration exposure to fibrogenic agents. And so what we did is we picked TGFbeta, which is a potent pro-fibrogenic cytokine, as our positive control at two different concentrations. So a concentration that is physiologically relevant and one that is traditionally employed in vitro model systems. And for our fibrogenic drug and chemical, we decided to use methotrexate at a physiologically relevant concentration, the plasma concentration, but also an estimated sinusoidal concentration, or what the liver would see, and also thioacetamide, which is a chemical commonly used in rodent models of liver injury leading to fibrosis. And so the study design was as follows. The tissues were bioprinted and cultured for 3 days before compound exposure. And we did daily treatments of each of these agents. And on alternate treatment days, we took non-invasive measures or samples from the media to perform LDH analysis, but also measure albumin production and cytokine secretion. And at the end of the treatment time course, we then harvested the tissues for RNA and also histology. And here are just graphs showing the impact of fibrogenic agents on some of the biochemical markers. On the left-hand side, you see the gross morphology of the tissues after 14 days of treatment. And you do start to see, as a result of treatment, some differences in the overall gross morphology of the tissues. And what looks like in some of these treatment groups, a consolidation and loss of tissue mass. And if we look over here at LDH release as a marker of injury, you start to see that during the initial time period for methotrexate that we don't mainly see statistically significant increases in LDH release. But over time, what you start to see is an increase in LDH release, suggesting we have mild to moderate injury occurring later on during exposure. And so if we look over here for the graph with thioacetamide, this trend is similar. However, at the 25 millimolar TAA concentration, you do see a monophasic increase in LDH. But for the 5 millimolar thioacetamide treatment, there is a similar trend as methotrexate over time. And if we look down here at albumin output, we do see that by treatment day 14 and also treatment day 7, starting at treatment day 7, that we see perturbations in hepatocellular function with some of these treatments. And we feel that this mild to moderate injury is what we would think would set the stage for a fibrogenic event. And so it was, excuse me. And so what was really striking was evidence of collagen deposition and the appearance of matrix within the tissue, which is shown here in blue. And so right here on the left-hand side, we have the vehicle-treated tissue. And so this is just a transverse cross-section of tissue where this top portion is the apical face of the tissue, and this bottom portion is the basolateral, which is the face of the tissue that would come in contact with the transluminal membrane insert. And so what you start to see with TGFbeta treatment is an increase in collagen deposition, more diffuse areas of collagen deposition within the non-parenchymal compartment, which makes sense because that's where the hepatic stellate cells reside. And so at the 10 nanograms per mil TGFbeta treatment, you start to see thickening at the basolateral edge of the tissue and increased collagen deposition denoted here with the arrows. And so for the methotrexate, you do start to see a dose-dependent increase in collagen deposition and pretty remarkable remodeling of the tissue, where you start to see nodular areas of collagen deposition and then areas in which the collagen bridges hepatocellular compartments, which is shown here with the yellow arrows. And then we start to see pericellular fibrosis, which is essentially collagen surrounding hepatocytes. And this is shown here in this little inset below. And so with the thioacetamide treatment, we start to see more pronounced fibrogenic features with more extensive whorls of collagen deposition and replacement of the hepatocellular compartment with these areas of fibrosis. And then we also start to see entrapment of these hepatocytes in this scar-like tissue. So this kind of gets back to the compartmentalized architecture of the tissue and how we're able to elucidate some of these more complex patterns of fibrosis that are more analogous to what you would see in a clinical specimen from a patient. So down here is just a clinical specimen from a patient that was treated with methotrexate. And to the right-hand side of it is the 1 micromolar methotrexate treated group. And what you start to see are patterns that are pretty similar to what has been described in vivo. And I'd just like to stress again at this point that histology remains a gold standard for assessing collagen deposition. In addition to this, we also decided to look at the prevalence of collagen subtypes that tend to be upregulated in fibrotic liver. So we picked vehicle control and 1 micromolar methotrexate and 25 millimolar thioacetamide, which were the treatment conditions exhibiting the most pronounced fibrogenic change, and looked at collagen 1, which is mainly localized to areas of fibrillar collagen, which I have outlined here in the 1 micromolar methotrexate treated group, but also in areas where you have pronounced collagen deposition, areas in which you have nodular fibrosis. And so collagen 4 as well was mainly localized to the periphery of the hepatocytes, and was also seen to be visually upregulated in treated tissues. So collagen 4 didn't really pick up some of these thicker collagen fibers, but you do start to see upregulation in some of these areas where you have nodular collagen deposits. And so we further looked at vimentin, which is a marker of the non-parenchymal cells, to just assess the distribution of these cells in response to compound-induced injury. And compared to vehicle-treated control, you see that these cells are more diffuse and exhibit a more punctate patterning. As with treatment, you start to see a more diffuse and concentrated areas of these cells, particularly in areas where you have nodular fibrosis and prevalent collagen deposition. And so these staining results further suggest to us that these tissues are undergoing sort of an active remodeling with treatment and very pronounced collagen deposition. Furthermore, we measured the abundance of pro-inflammatory and chemotactic cytokines secreted into the culture media. And so over time, we do see an increase in pro-inflammatory IL-6, suggesting these tissues are in an active phase of wound healing. And also, we start to see the upregulation of chemotactic proteins, but also differences in immunomodulatory cytokines such as GM-CSF. And also, we start to see transient increases in different patternings at different time points during the exposure. And these results were further supported by gene expression, looking at alpha-smooth muscle actin, which I had mentioned before is an activation marker of hepatic stellate cells, but also collagen 1A1. And we start to see by treatment day 14 that we start to see upregulation of these transcript levels in treated tissues, further suggesting that these tissues are in an active state of wound healing. And so I just would like to say in summary that these XV3D bioprinted liver tissues represent a unique platform for measuring some of the effects of repeated compound exposure. And so this novel model system may lead to a better understanding of some of these early key events underlying chronic injury, and also classification of fibrogenic agents for risk assessment purposes. And so as a additional application, these results will also help inform effective therapeutic approaches for intervention at both early and later stages of the disease, for which different cell types may be involved. And so in summary, these collective results show strong evidence that bioprinted tissues can recapitulate some of the cellular, molecular, and histopathological events associated with drug, chemical, and TGFbeta induced fibrogenesis. And so I'd just like to thank my funding and support, and I would be happy to take any questions.
Thank you, Leah, for a very nice seminar. We'll now begin the Q&A segment. As a reminder to the audience members, you can still submit questions through the question pane in the attendee control panel. So with that, let's get started. We did get a couple questions about Kupffer cells, so I'll break those up. First, just simply, does the model contain Kupffer cells, and if not, why were they excluded?
That's a great question. So the standard model system that we evaluated does not include the Kupffer cells. So one of the advantages of this tissue model system is that you can actually customize the different cell types that go into the liver model and also the ratio of cells. So in the future, we can further assess what the role of Kupffer cells are by either specifically adding them or taking them out of the culture system in a controlled fashion.
And a related question, if you had included Kupffer cells in the fibrosis experiments, what would you expect for a result?
So that's another good question. So given that Kupffer cells are the resident macrophages in the liver, and your liver does undergo insult on a daily basis, we would hypothesize that the Kupffer cells would help modulate fibrosis with more of a homeostatic function, and that if we were to incorporate Kupffer cells, we might be expected to see a delay in the progression of their response.
So next question, what is the background rate of fibrosis without drug treatment in the model? In other words, how much fibrosis occurs over the course of 28 days in a vehicle control?
So I have not carried out the tissues to 28 days in vehicle control, but you did see a 14-day exposure. And even in the vehicle-treated control, you do see some areas of collagen deposition, which are normal, typically at the basolateral edge of the membrane of the face of the tissue in contact with the transwall membrane, but very minimal collagen deposition throughout the tissue construct. So I would expect at 28 days you would still see hints of collagen deposition, but typically not to the extent as you would in a treated tissue.
Okay, next question. Given that fibrosis can take months or even years to develop clinically, why do you think you're seeing evidence in the model of fibrosis in a 2-week time frame?
That's a good question. So we're not really sure why this particular phenotype is accelerated in this model system. And so one of the things that might be a factor is that we don't have blood flow or circulation in our model. And so that some of these clearance mechanisms that might be operating in vivo that might slow the progression is not represented here in the model. And also Kupffer cells, that as I had mentioned before, may have a more homeostatic role in vivo that might be expected to modulate progression.
Okay, next question. Can you recreate the recruitment of inflammatory cells in this model system?
Yes, so we haven't looked at that yet, but it's a very interesting experiment that we could potentially perform. and we could directly incorporate different immune cells into the tissue, and because of the compartmentalized architecture, we could actually see the migration of the cells throughout the tissues. So that'll be a very interesting experiment to perform in the future.
Okay, next question. Do you see donor variation in the fibrogenic phenotype with this model?
So, so far with the couple of donors that we've tested, the responses that we see are pretty consistent. But in order to get a real feel for some of the variation in either the progression or the susceptibility to hepatotoxicity following exposure to these compounds, we would need a larger population to better understand those differences.
Okay, here's a very good question. Have you seen reversal of fibrogenic phenotype after removal of the inducer, or have you been able to block it with an antifibrotic compound?
Yes, that is a very good question, and that's something that we actually have planned for some of my upcoming studies. And so what we're specifically interested in is looking at the impact of how the degree of injury to the hepatocytes might affect the reversal after you remove the etiological agent. And so there have been studies in the literature suggest that fibrosis is a reversible process, but depending on how far gone, whether you're into full-blown cirrhosis, there's either slower reversal or not much reversal at all. So once we kind of understand some of those processes leading up to fibrosis, but also the reversion, we can look at some interventional strategies using some of these antifibrotic treatments that are available, and kind of get a better understanding of the temporal changes that are occurring and when there would be a good time point at which you could introduce an agent for it to be effective.
Okay, another question. What do you think is driving the bridging fibrosis that you observed? Does it have to do with the proximity of two focal fibrotic regions within the tissues?
Yes, so pretty much damage to the parenchymal compartments adjacent to those non-parenchymal regions will facilitate or attract cells to the site of injury, so that migration of stellate cells bridging from one compartment to another could be possibly explained by that.
So another kind of set of related questions is what happens to the tissue after bioprinting? Do the cells proliferate or move around, and then is it different in fibrogenic induction experiment?
Yeah, so the tissues do mature over time, and you do see some proliferation of the non-parenchymal cells, but pretty much the cells stay within distinct parenchymal and non-parenchymal compartments, and these are well defined over time. But when you do treat with fibrogenic agents, as you saw in some of the histology cross-sections, the increased collagen deposition and proliferation of the non-parenchymal cells, in addition to injury to the hepatocytes, ultimately leads to a distortion of that architecture and pretty much contraction of the tissue.
Okay, it looks like that's all the questions we've got, and I do want to thank the audience for your time and for the excellent questions that were submitted. From Organovo, please look for our follow-up email. To those of you who registered for the webinar, it'll contain a link which allows you to review the recording of today's webinar, and that will be available on our website, organovo.com. I would also like to point out that we offer the human liver fibrosis model through our tissue testing services, so if you're interested in exploring this further, please do check out our website. So on behalf of Leah Nerona, as well as the team here at Organovo, thank you for tuning in, and have a great rest of your day, everyone.